Hello there and welcome to this webinar on alternative approaches to mammalian acute toxicity testing, which is the fifth webinar in our series of alternative approaches, which is co-sponsored with the PETA International Science Consortium. I'm Emma Chenoweth of Chemical Watch and I'll be moderating the event. Introducing the speakers today, we have PETA's Dr. Amy Klippinger. Before I hand over to Amy, I'll just explain how the webinar works for those of you who haven't joined us before. Today we have two speakers who will present together for about 40 minutes, and then we'll go to a Q&A session which will last for possibly about 20 minutes or so. So we aim to be around about an hour during this webinar. The delegates can ask questions during the webinar by using the questions box, which you'll see. I can see it's on the right-hand side of my screen, but I think it's on the right-hand side of your screens. So you can use that to ask questions, which if you ask them during the webinar, I can forward them on to the speakers. So if you can do that in good time, they'll have a chance to look through them as we go along. And if your question's not answered during the webinar, and sometimes we do get many questions, so you'll forgive us if we don't get round to them all, but you can use our forum to submit them to, and then we can address them after the webinar. And we provide a link to this forum towards the end of the webinar as well. Now, if you experience any technical problems, I know sometimes people have difficulty with sound and things like that, but you can send a query through to Lorna, my colleague, who should be able to assist you if you do have any problems, so you can do that. One of the most common questions we get is whether we're going to produce a recording of the webinar, and of course we will, along with the slides. That's normally available a few days after the webinar, and it's posted on the Chemical Watch and also the PETA International Science Consortium websites. I think we also send them out to you, so they'll definitely be on their way. Right, now, without further ado, let me hand you over to Amy, who's going to provide you some information on the webinar and also introduce the speakers. Thank you, Amy.
Thank you, Emma. I'd like to also welcome all the participants to today's webinar, which is focused on alternative methods for acute systemic toxicity testing, and to thank Chemical Watch for their support in hosting these webinars. The goal of this six-part webinar series is to help registrants plan for the 2018 REACH deadline. Hundreds of thousands of animals have already been used for REACH testing, and millions more are expected to be used in the coming years. However, there are alternative methods that can be used to meet REACH data requirements while minimizing animal testing. For more information on minimizing animal testing for REACH and for recordings of past webinars, please feel free to visit our website, and also feel free to get in touch with any questions after today's webinar. As a quick reminder, the sixth webinar in our series, which focuses on alternatives to fish acute toxicity testing, will take place on April 14th. Today, our speakers are Drs. Pilar Prieto and Larry Milczek. Dr. Prieto coordinates activities related to acute systemic toxicity at the European Union Reference Laboratory for Alternatives to Animal Testing of the European Commission Joint Research Center. This organization promotes the scientific and regulatory acceptance of alternative methods. Dr. Milczek is the Senior Manager of Toxicology and Strategic Services in the Corporate Medical Department at the 3M Company. Here he manages the work focused on the development and use of in vitro techniques and directs the Computational Toxicology Assessment Program. For the first 30 minutes of the webinar, Dr. Prieto will present on ECVAM's recently released strategy to replace, reduce, and refine the use of animals in the assessment of acute mammalian systemic toxicity. If you haven't downloaded it already, this report can be found on ECVAM's website under Strategy Papers. Following Dr. Prieto's talk, Dr. Milczek will speak for about 10 minutes on industry experiences in using alternative strategies to meet acute systemic toxicity testing requirements. With that, I'll pass the controls over to Pilar to start.
So thank you, Amy. Hello, everyone, and thank you for participating in this webinar. So here you can see the outline of my talk today. I will start with a brief introduction of the regulatory in vivo tests that are used today to assess acute systemic toxicity. I will then provide you with an overview of the regulatory framework in the EU. Next, I will go through some mechanistic considerations for this human health endpoint, as well as an overview of efforts to develop non-animal methods for acute systemic toxicity, and then I will end my talk by introducing the EURL ECVAM strategy on acute systemic toxicity. Next slide, please. So, acute systemic toxicity describes adverse effects that results from single or multiple exposure to a substance or mixture within a maximum of 24 hours. Following the exposure, the toxicity is typically investigated for a period of at least 14 days. The substance can be administered orally, dermally, or by inhalation. And today, all the accepted methods are done in vivo according to the OECD test guidelines that are shown here in the table. So by the oral route, there are three sequential dosing methods that are, in fact, modifications of the classical LD50, and therefore they are refinement and reduction methods. By the dermal route, there is only the classical LD50 test available, and via inhalation, there is a revised version of the classical test method, as well as a refinement and reduction method that is the acute toxic class method. Most of those standard in vivo tests use death or imminent death as the apical endpoint, all except the oral fixed dose procedure that is based on the observation of clinical signs of toxicity, so evident toxicity. So next slide, please. Next, please. Okay, thanks. Data from acute systemic toxicity are used primarily for classification and labeling purposes, and the table that I'm showing here shows the categories for classification for the three routes of exposure according to the EU CLP system that implements categories 1 to 4 from the global harmonized system. In the table are shown the LD50 and LC50 values that define the category boundaries for each route of exposure, together with the pictograms, the signal words, hazard statement, and also summary of precautionary statements used for labeling purposes. It is worth here to note that the same pictogram and signal word is used to communicate the hazard for categories 1 to 3, and the hazard statement for categories 1 and 2 is the same, fatal, and different from category 3 and 4. Since the information that obtained from these studies are hardly, if ever, includes any target organ effects or mode of action or toxicokinetic data, the usefulness of the data that are obtained for other purposes than classification and labeling are limited. However, we go to the next slide, please. We have to say that, the next please, the information may also be used to support elements of risk assessment and risk management, such for instance the setting of occupational exposure limits or clinical emergency response planning. And here at present in Europe we don't have a broad consensus available on guidance for risk assessment, risk management, risk communication purposes in cases of chemical incidents. The REACH regulation and guidance document do not specifically address emergency situations, and what the guidance says with regard to the establishment of an acute systemic DNL is that it is problematic in a way, and in general is not necessary, since the long-term DNL is normally enough to ensure that these effects do not occur. However, in this guidance also says that it is required, this acute DNL, for volatile hazardous substances when there is a potential for peak inhalation exposures. Next slide, please. Here in this slide, I provide you with an overview of the regulatory framework in Europe, and it is clear from the slide that acute systemic toxicity is a common requirement for the majority of chemicals across sectors, and the information requirements vary depending on the type of chemical that is under regulation. According to the EU CLP system, all marketed substances and mixtures must be classified and labeled irrespective of the quantity placed in the market. This system per se is not setting information requirements, and the classification must be carried out on basis of available information. Acute systemic toxicity is a standard information requirement for all chemicals that are registered under REACH, and here the data are required by a single route of exposure or by two routes of exposures depending on the tonnage level. For biocides and plant protection products, the data are required independently of tonnage. For biocides, the selection of the second route of exposure depends on the nature of the substance and the human exposure. In the case of the plant protection product regulation, there are specific criteria and conditions specified when the dermal and the inhalation routes may be required. For cosmetics, acute toxicity is part of the minimal base set requirements for ingredients that are listed in the annexes of the regulation, so for those that are of high concern for human health. Nevertheless, for these in vivo acute systemic toxicity studies are prohibited for cosmetic substances and products in Europe. We have then also a sector, that is the pharmaceutical sector, that does not longer require acute systemic toxicity studies by default, since it has been shown that as standalone tests they are not useful in supporting the first clinical trial in humans, and they also not provide additional value in supporting the management of overdoses. And in fact, the information needed on acute toxicity can be obtained from any other short-term or dose escalation study that in a way is performed as part of the development process. So if we go to the next slide, please. Here, we coming back to REACH, we can see that under Annex 7, there are specification of the standard requirements for substances that are manufactured or imported in quantities of 1 ton or more. Okay, so we start then with the mechanistic aspects that I wanted to mention. I was saying that lethality is considered to be the result of non-specific interactions of the chemical with those structure or processes that are vital for cell survival, proliferation and function, so what is known as general basal cytotoxicity. And these non-specific interactions may in the end lead to organ tissue-specific toxicity and lethality, and this is something that may hold true for the majority of chemicals, but it is also recognized that a certain number of chemicals will produce cell-specific toxicity. And for instance, the tables that I'm showing here are the results of the discussions on an ECVAM workshop on strategies to replace acute systemic toxicity. So the table on the left side is showing mechanisms common to many cell types responsible for cellular failure or death, while on the right part we had organ-specific functions that are commonly compromised in organ failure, and the table lists the main target organs that are known to be susceptible for acute toxicity and that in the end are resulting in lethality. And I was saying that it's obvious from the table as well that there are some physical processes like transportation of molecules, membrane integrity and secretion of molecules that are common to many organ systems. So if we go to the next slide, please, we have another table that sums up the issues that were discussed in another follow-up workshop on acute chemical safety, and in this table we have key processes associated with acute human poisoning that were listed by a number of pathologists that participated at that meeting. And during that meeting it was also very clear said that the mechanistic information could be very important for developing more predictive in vitro test methods. But even all these efforts, today we still have a poor characterization of modes or mechanisms of actions next for acute systemic toxicity in humans. So next slide, please. Then, of course, the toxicokinetic factors are also very relevant and important since we are talking about systemic effects, and cannot be ignored in any of the testing strategies or approaches that we may need to put together for acute systemic toxicity, together with the assessment of basal cytotoxicity, cell-specific toxicity, and cell-specific function. And here I wanted to emphasize also that we have drafted recently a strategy document on toxicokinetics, which aims to identify opportunities for generating and making a better use of toxicokinetic data. And this document will be hopefully finalized in the course of the year and made also available through our website. Next slide, please. Okay, now here we go to what has been done in this area for developing non-animal methods. And here I was saying that if we take into account that for the majority of the chemicals lethality is considered as a result of general basal cytotoxicity, then it's really possible to to think that the prediction of acute systemic toxicity in vitro using non-specific effects as endpoints would be a possibility. And this is in fact the rationale that has been used for using cell-based methods, and in particular in vitro-based cytotoxicity, for the prediction of acute oral toxicity in a number of studies, international studies. And here in this slide I'm just showing the major ones. We have the MAKE program that was assessing the relevance of in vitro cytotoxicity tests for human lethality. We have the HALE register of cytotoxicity, that is a database of published cytotoxicity values and rodent LD50 values. And then we have the international validation studies of the neutralized uptake. All these studies have shown that there is a general good correlation between in vitro cytotoxicity data and oral LD50 values, and this indicates that in fact basal cytotoxicity is an important event in acute systemic toxicity. The validation studies also show that these methods, however, they are not accurate enough to replace the in vivo studies for acute oral toxicity. However, they could be used to support the selection of the starting dose for the in vivo test, and this is in fact described in the OECD guidance document 129. The fact is that those approaches have not been really taken up in practice, and the contribution to the reduction in the animal numbers have been recently questioned by some authors. But we also have evidence that these cytotoxicity assays showed in general better predictivity for substances of low toxicity. And in this regard, ECVAM has issued a recommendation on the use of the 3T3 to support the identification of non-classified substances. Then the role of kinetics organ specificity in combination with cytotoxicity assays was evaluated in the EU research project Acute. And then we also have a range of cursor models that have been developed to predict acute systemic toxicity, either the LD50 values or the classifications, and all of those methods are statistically based methods, but they are also predicted for specific chemical classes. Next slide, please. Here I just wanted to come back again to the 3T3 neutralized cytotoxicity assay and to talk a bit more about the ECVAM recommendation that was issued following the validation study that aimed to evaluate the capacity of the assay to specifically identify non-classified chemicals. Then we have recommended the use of the assay as a part of an integrated approach that could be a weight of evidence or an ITS for supporting the identification of non-classified substance when the threshold of 2,000 mg per kilo is used. However, the method has limitations, mainly with regard to the detection of a specific target organ toxicity and the lack of assessment of metabolites, and therefore the assay has to be used as part of an approach that also includes the assessment of other data sources, including here read-across, physical properties, structural layers, toxicokinetic data. The assay, however, is particularly relevant for the assessment of industrial chemicals, which tend to be of low toxicity. However, even in this case, care must be taken when we interpret the results in case the chemical is metabolically activated or detoxified. With regard to positive results and the fact that the assay has low specificity, this means that there is overprediction of many negatives as positives, so the positive result cannot be really used per se for the identification of the different toxicity classes. Next slide, please. Here, I just wanted to briefly mention some of the main findings of the Acute research project that aim to develop a non-animal testing strategy to predict human acute oral toxicity. The figure is showing the general structure of the project that evaluated and combined cytotoxicity, organ-specific toxicity assays, and also biokinetics and metabolism, and proposed a number of testing strategies to classify chemicals into official acute oral toxicity categories. Then the outcome with regard to the acute oral classification of substance supports the use of the 3T3 neutralized uptake within a tiered testing strategy to identify non-classified chemicals. However, the classification analysis that was carried out also confirmed or showed that by complementing the cytotoxicity assay with additional endpoints did not really provide a significant improvement in the classification of compounds into categories 1 to 4. In addition, also, all the 4 proposed testing strategies have a tendency to overpredict toxicity. But what the project also showed is that the target organ-specific in vitro assays proved to be useful for identification of alerts for specific toxicities, for instance neurotoxicity, and within the project an approach was suggested that combined the prediction results gained with a 3T3 neutral assay for the identification of negatives with neurotoxicity alerts, and in this way it was possible to reduce the number of chemicals wrongly predicted as non-classified. Next slide, please. Okay, thanks. So now, with talking about inconsistency of classification of any alternative approach, it's important also to reflect on the precision of the established methods. One slide back, please. Here. Okay, so this graph that I'm showing here is just the results of an analysis of consistency of classification that was performed within the AccuTox project. The graph is showing the numbers of chemicals that are falling into 1, 2, 3, or 4 classification categories, and here we show both the EU CLP system and the global harmonized system according to the LD50 ranges that were collected for the reference chemicals. And the analysis what showed is that with at least 90% probability, 50% of the substance would fall into only one toxicity category, while around 40% of the substance would fall within 2 neighboring categories. So it's important then the misclassifications also always to be interpreted in the light of the imprecision of the in vivo method. Next slide, please. Here I just wanted to complete this overview on non-animal methods, just briefly mentioning the results that we obtained in an in-house study that aimed to evaluate whether the combination of Kussar models with cytotoxicity could provide an improvement into the prediction of acute oral toxicity. So there were 4 Kussar models evaluated together with the 3T3 neutral uptake and was applied to a set of 180 chemicals, and the performance of each method alone and in different combinations and stepwise testing strategies was evaluated. So what the main outcome of the project is that 2 Kussar models, the ones shown here, were the best performing when used individually in terms of predicting the LD50s, but also the toxicity classes. The 3T3 neutral uptake showed the lowest accuracy, and then when the 5 methods were combined in different combinations, in fact there was not any real improvement of the predictive performance, but here you have the possibility to combine in different ways to maximize sensitivity for toxic chemicals or specificity for non-toxic chemicals. So next slide, please. So with this overview, it's evident that there is a need to develop and validate alternative methods, more integrated approaches to satisfy regulatory requirements for acute systemic toxicity. And this is in fact why we have published recently the strategy document that has an ultimate aim to propose solutions that can satisfy regulatory requirements under several pieces of EU legislation. We have a number of objectives under 2 strategic aims that are intended to progress the field on multiple fronts, but we also, with that, we want to have a framework that help us to prioritize methods that are submitted for us for validation. So, next slide, please. Here, this figure, previous one, please. Okay, so this figure is just meant to give you an overview of the different objectives that we identify under the 2 main strategic aims that we had, and also you have in the slide the link where you can download the document. So if we go to the next slide, please. One of the objectives that are identified in this document is the development and the optimization of mechanistically relevant alternative methods for use within integrated approaches for testing and assessment. The fact that, as I said today, we are still lacking a complete mechanistic understanding of the numerous toxicity pathways and mode of actions that lead to acute systemic toxicity may in a way hamper the development of IATA for this endpoint. And therefore we think it's important that we improve the theoretical understanding of acute systemic toxicity so that we could develop mechanistic relevant alternative methods which in the end, together with cytotoxicity assays, are expected to contribute to the weight of evidence in future IATA for this endpoint. In the meantime, and ECVAM is also proposing to explore options for making better use of existing alternative methods. One could be the neutral cytotoxicity assay, and for instance, understanding the reasons why we have false negative results, trying to identify the relevant and complementary information that could help to interpret the negative outcome from the study. Next slide, please. So, important also, information from repeated dose studies, if available, could be also very useful in supporting classification and labeling for acute oral systemic toxicity. And in this regard, we have done some preliminary work in the past with the aim to identify non-classified substance from the results of a 20 days repeated dose study, where we identify an OL of 200 milligram per kilo, above which we could correctly identify around 63% of the non-toxic compounds, while still we have a small percentage of harmful compounds misclassified as non-toxic. With this in mind, would be relevant then to further explore the use of data from these repeated dose studies in order to gain information on toxic effects that could in the end support the classification and labeling. And this would be in fact very relevant now for the REACH 2018 deadline for substance under Annex 8, for which the repeated dose data are required. We have launched recently a survey that was aimed to gather additional information and expert opinion in the field, and in particular was the aim was to get additional insights on the possibilities to conclude on acute systemic effects from the systemic repeated dose studies. And now I just wanted to say that ECHA is currently investigating whether information from repeated dose studies can be used by registrants to adapt their information requirements related to acute oral toxicity, also in combination with the in vitro cytotoxicity data. And here the intention of ECHA is to ensure that all the relevant guidance documents are updated by 2016. So next slide, please. Next, okay. With regard to the development of waiving arguments to avoid animal testing, I would like to mention here the response of the competent authorities for REACH and CLP, Caracal, to the proposals that were received for modification of the REACH standard requirements for acute toxicity in Annex 8. From the several elements contained in these proposals and that are listed here in the slide, they have agreed to amend the Annex 8 so that substances that have not shown acute oral toxicity up to the limit of 2,000 milligrams per kilo would not require the dermal data. The other technical aspects of the proposal should be included in an ECHA guidance rather than in the Annex amendments. We also have an OECD project that aims to develop guidance for waiving and bridging mammalian acute toxicity test, and here including the acute systemic toxicity for pesticides and biocides. And this project gives opportunities for collaboration between different stakeholders. Next slide, please. Another important objective in the document is that whenever an animal test is still needed, death should be avoided and more humane indications of toxicity should be used, as this is something that is encouraged by the EU Directive 2010/63 and also by the OECD Guidance 19. As I said at the beginning, today only the oral fixed dose procedures uses evidence signs of toxicity to provide the range estimates. And in line with that, we have now two ongoing activities in Europe that are supported by ECVAM. One is a project of the European Partnership on Alternative Methods that is focusing on building a decision framework document to replace animals in acute toxicity testing. And if the animal cannot be avoided, the idea is to replace mortality by clinical signs that are predictive of mortality at a higher dose level. This framework is also envisaged to use in vitro cytotoxicity as well as clinical signs from range-finding studies as part of a weight of evidence assessment. And then in parallel, we have another activity that is led by the NC3R Center in the UK that has as an aim to refine the inhalation studies through the use of evident toxicity as an endpoint. The ultimate aim here is to support the acceptance at OECD level of the fixed concentration procedure for acute inhalation. They have collected clinical signs during inhalation studies in rats, and they have used the data to develop a revised test guideline with clear, defined, evident signs of toxicity. During the analysis, and I listed here the clinical signs that have been identified as highly predictive of severe toxicity of death at the next highest dose. Between these two projects, there is a crosstalk in order to avoid duplication of work, and the EPA project is focusing then on the dermal and the oral route. Next slide, please. Within this strategic aim of refinement, we also have a new OECD project that is aiming to revise or either replace the OECD test guidelines 402 for the acute dermal toxicity test, and to put it in line with the 3R principles to refine the test and to reduce the number of animals. So, and with that, if we go to the next slide, please, I come to the summary of the presentation. So, at present, only the data derived from animal tests are accepted by regulatory bodies. However, the in vivo acute systemic toxicity studies are prohibited in Europe for cosmetic substances and products, and furthermore, under REACH, tests on animals must only be conducted as a last resort, and in the Annex 11 is describing how the standard requirements can be adapted. The strategy document proposes that the effort should be directed towards the reduction and replacement of animal tests for the identification and classification of acute systemic toxicity, the use of in vitro cytotoxicity assays as additional tests for estimating the initial doses for the test in vivo is described in the OECD guidance document. There is also evidence indicating that the 3T3 neural uptake can be used to support the identification of non-classified substance when we are using the threshold of 2,000 mg per kilo. However, due to the limitation of the test, the results should always be used in combination with other information sources. The information provided by mechanistically relevant in vitro assays, as well as existing information on repeated dose studies, is expected to contribute to the weight of evidence in future integrated approaches to testing and assessment for this endpoint. And also, last but not least, whenever the animal test is still needed, the effort should go into the refinement of the in vivo study, and in particular to avoid lethality as an endpoint for acute systemic toxicity testing. And with that, I conclude my presentation and I hand over to Larry.
All right, thank you, Pilar. That was excellent. So I was asked to provide some perspective on our experiences here at 3M and some of our initiatives around looking for alternative strategies for acute toxicity testing. So I realize we're running maybe a little tight on time, so I'll do my best to move through my brief presentation pretty quickly here. We'll start with a little bit of background on the group that I'm in. We are in the 3M Corporate Medical Department, and I'm managing the Toxicology and Strategic Services Group, which basically includes all of the corporate toxicologists here at 3M, providing support to all of the 3M businesses. That also includes our computational toxicology group, and also our on-site toxicology laboratory that we call the 3M Strategic Toxicology Laboratory. Next slide, please. One of the interesting things that we deal with here at 3M is, obviously the 3M product line is very vast. 3M is represented in a number of different markets, including healthcare products, consumer products, industrial products, chemicals, and so forth. So the business needs that we have in terms of acute toxicity testing can be highly variable and highly specific for an individual business. So these types of needs can range from everything including GHS classification, EU classification for the purposes of hazard communication documents. We can also have data needs around new chemical registrations, various different types of regulatory submittals to governmental and occasionally non-governmental agencies, as well as basic risk and safety assessments. And quite often, these types of tests are needed for basic raw material selection and raw material screening. So when we look at the company and our group as a whole, we've got a lot of different types of needs and applications potentially for acute toxicity test data. Next slide, please. So one of our basic goals here at 3M within the medical department is to try to avoid the classic, animal-based acute toxicity testing where possible. And I'll be quite honest, obviously that's not always possible. There are still various regulatory requirements throughout the world that will mandate that type of testing depending on the application. But what we're trying to do is come up with some internal processes where we're using a combination of computational as well as laboratory methods to try to reduce as much of that testing as possible. Next slide, please. So I'm going to start by discussing our basic approaches from a computational perspective. Generally, what we have found is for our chemistries and the types of assessments that we are performing, it's most effective if we rely on more than one method, more than one type of approach, and in essence use a kind of a weight of evidence philosophy in terms of computational toxicology. In general, we try to utilize a three-tiered strategy for our assessments, and that's utilizing classic QSAR programs, number one. Number two, reviewing compounds for structural alerts that may be associated with particular types of endpoint toxicity. And then number three, read across, in essence where we're looking for structural analogs of our compound of interest that may have available toxicity data in the literature. And what we have seen is, although there often are inconsistencies and differences between the approaches, we also have a great deal of increased confidence in our end result, our estimates, and our conclusions when we are seeing agreement among the three different approaches. And whenever we utilize these three different approaches, particularly on the QSAR programs, we don't necessarily just run the program, provide an input, and then take the output at face value. We try to add a lot of expert judgment and hopefully, professional history in terms of how we're utilizing these programs. And from our perspective, we think that adds a lot of value to the process. And again, it really goes a long way towards increasing our confidence in these types of assessments depending on the application it's going to be used for. Next slide, please. Generally, when we're looking specifically at acute toxicity, primarily what we're looking for are a way to classify the chemical or compound of interest into an approximate GHS, acute toxicity classification range. And for the most part, we feel that we've developed a process where we can be reasonably effective at that. And we've done this quite a lot since about 2009 when we fully instituted this process. Now, with that said, we do have a lot of limitations, and I think we've learned a lot about where this type of process can be most effective, and we've also learned about where the process might be somewhat inappropriate. So we try to be very judicious in how we are applying these types of techniques. We are using a combination of free and subscription software to run the QSAR type pieces. And we're also working on building QSAR models based on 3M historical test data. And that's something we've been initiating a few projects on over the last few years. And we think that can actually have a great deal of success for us. And we're actually trying to look at how we're running other studies moving into the future so that perhaps we can generate data that's also going to be more useful to building these types of computational models into the future. Next slide, please. What are some of the challenges that we've seen specifically for acute toxicity? Well, for the types of chemistries that 3M is involved with, very often these chemistries are not necessarily well represented in commercial QSAR programs. That's definitely an issue. And again, we feel that's where our professional judgment and expertise in when to rely on these types of predictions and estimates is really quite critical. And also, when we're talking specifically about acute toxicity, structural alerts may in and of themselves not necessarily be particularly effective. They can be more effective for other types of endpoints that we may be asked to assess, but in terms of acute toxicity, in some cases they may have limited utility. As I already mentioned, we do routinely see inconsistent results, lack of correlation between the different approaches. But we also definitely have cases where we see good consistency, and we see agreement among the different approaches. And then that's where we start to have, again, much more confidence in those types of assessments, particularly with how we're going to apply the data moving forward. Next slide, please. What we're doing with respect to some laboratory techniques to potentially supplement our computational tox approach, and in some cases operate somewhat independently of the computational tox approach. But we have started to work with the guidance document that Pilar was talking about, OECD guidance document 129, the neutral red assay. And, I will tell you honestly, we have seen some limitations for the types of chemistries that we've looked at. Solubility can be a big issue. We've got some chemistries that have a certain degree of water reactivity. That can be a problem because this is a classic cell culture approach. But we also have seen some success stories as well. Many of the chemicals that we have looked at, when they are meeting the physicochemical requirements for the assay, meaning the physical property requirements in terms of solubility and reactivity, we actually have gotten very good results with this assay. And it's something that we're looking at, potentially trying to figure out how we can really build on this moving into the future. Excuse me. Another thing we're actually doing internally at 3M is looking to make a significant investment in terms of inhalation acute toxicity. And we're actually purchasing a system that's going to allow us in our laboratory to try to build some screening models using three-dimensional respiratory tissues and trying to look at correlations with acute inhalation toxicity. And this will be brand new technology for 3M, but it's something that we're very excited and interested in doing in the coming years, because we think it's really important in terms of furthering the science, but also building more robust processes for us internally here at 3M. Next slide, please. So in summary, I'll try to wrap it here. I see we're just about out of time, but here at 3M we're trying to be very proactive in what we're doing in terms of investigating alternative methods. We do believe that these methods have been useful for 3M as a company. In general, we believe that the processes we have built have helped reduce animal testing, but also from a corporate perspective, they've been successful in helping us screen chemicals more quickly, get information back to our business units that we're supporting in a more timely manner without the need to do animal testing. And for that reason, 3M is going to continue to invest in these types of technologies, and we hope to further refine our processes moving ahead. And I think, even though I mentioned that there are some limitations and some difficulties with these types of approaches, because we have seen success, it's something that 3M is going to continue to work on and continue to hopefully validate and improve moving into the future. Okay, so that concludes my portion of the talk. At this point, I guess I'll turn it back over to our organizers.
Thank you very much, Larry, and thank you very much to everybody for persevering there, and big apologies for the technical problem. I am now going to go to the Q&A session. I should just explain that under normal circumstances, I would have been looking at all the questions as they've been coming in through the webinar, but I was aware that that would have meant that you would have seen my mouse flickering all around your slides and the screen, so I haven't actually done that. So the question and answer session will be, I was just going to say, just bear with us. If you want to stay and listen to the Q&A, then please do. We'll try and run about 15 minutes of Q&A. But you'll have to bear with us as I actually go through and read the questions for the first time myself. So if you're happy to do that, then we'll get going with some questions. Actually, I saw one that was coming through just on the screen, which was called Pilar, and I think it was referring to I think a comment you made about using the alternative approaches to identify mechanistic routes, but the question was something like, how do you do that when you don't know what the mechanism is? I guess it's a chicken and egg type question. Does that make sense? Because I haven't actually been able to get into the questions yet. But,
I don't know if he's referring to the results that I was mentioning in relation to the Acute Tox project, that I said that we had seen the utility of some of those more mechanistic in vitro assays, in particular neurotoxicity assays, that we were using the brain aggregates, that these are the 3D models, and they were able to identify by looking at selected genes some neurospecific effects, but also other more general effects, more stress-related effects. And then when you compare the LOEC values obtained in those in vitro assays compared to the IC20 values obtained with a 3D3 assay, you could decide whether there is an alert coming out from this in vitro neurotoxicity assay that could tell you, okay, maybe your negative result in the 3D3 is not negative in fact, and there is some potential for target organ toxicity. So I don't know if the question was exactly that.
Yes, I've actually found it now. It says, how do you decide what assay is mechanistically relevant when you don't know the mechanism of action?
Well, I think, yeah. Yeah, I think this is a good question, but in this particular case, I mean, well, this was done with the compounds that we use in this research project, and of course they knew the compounds that we were evaluating, so they knew that those compounds that were coming out as alerts, in fact, could have some neurotoxicity. But when it's an unknown, yeah, I mean, this is difficult to say.
Okay then, and there's another question here to do with one of your graphs. I think it was the correlation graphs between the in vivo and the alternative methods. The delegate has noticed that it had a scale of 0 to 5 grams per kilogram, he thinks. But he's noted that the EU only requires classification up to 2,000 micrograms. So he's saying much of the correlation on the graph isn't relevant to EU classification. Is that correct?
I don't know which slide he's referring to, that I showed correlations.
Have a look.
I don't know if he's...
Maybe it's a bit of a technical question. Maybe it's something that he can follow up with you afterwards.
Yeah, maybe we can look again better to the question and provide an answer in written.
Yeah.
Because I'm not sure exactly which slide he's referring to.
Yeah. Okay then. And there's a question here about whether or not there are any in vitro tests that can be done in lieu of the acute toxicity pathogenicity studies required by, as she says, for the EPA. So maybe this is not fair to ask you this, Pilar, but for microbial pesticides, the question is. I don't know if you know about the EPA.
Sorry, I cannot.
Okay,
I can think about it, but I cannot provide right now an answer for that.
Yeah. Yeah. Okay then, well we can see if we can follow it up afterwards maybe. I've got a question here for Larry, and it's about whether or not he can disclose more information about the 3D inhalation model that 3M are looking at.
Yeah, so I can tell you we're in the process of purchasing a piece of equipment. It's called a vitrocell. And the vitrocell is going to allow us to expose tissues in culture, three-dimensional human tissue cultures, to gases and vapors as a starting point. And we hope to begin that work this year, in fact.
Okay then. I had a couple of questions for you myself actually. When you were talking about the computational approaches, I wasn't clear whether or not you were developing them for kind of chemical groups or for specific endpoints within 3M.
Both actually. This presentation was focused specifically on acute toxicity, but in essence we've got a computational process here within our group that will look at a variety of different endpoints, from dermal irritation to dermal sensitization, acute toxicity, genotoxicity, and so forth. So we have developed specific approaches based on endpoint, and then we've also we're basically looking at different chemical classes that 3M is interested in terms of developing chemistries and developing products, and then building specific models around those classes for the individual endpoints. Yeah.
And on that, I guess further development is, how do you then bring that work out into a kind of regulatory context? Do you have your work kind of peer reviewed by experts in this field, or how do you go about actually gaining regulatory acceptance for these kind of approaches?
It really depends on how the test data, how the computational data is going to be applied. A lot of the assessments we're doing are screening level assessments where we're looking at multiple chemistries that could be of interest to a business. So we're looking at maybe some compounds that have a better profile or appear to have a better profile. In other senses, what we're doing is really combining that three-tiered approach where we're looking at QSAR, structural alert, and read across, and where we see good correlation between the three approaches, then we have pretty good confidence that the overall conclusion is likely to be correct. From a regulatory perspective, oftentimes its best use is as supplemental information at this point. But it depends a little bit on the exact application. Again, I've mentioned our needs here are highly varied, everything from healthcare to consumer products to industrial chemicals. So we've done a lot of different types of assessments in this group with a lot of different endpoint applications. So I guess the overall conclusion there, it really depends. We really try to customize our approach based on how we believe the data is going to be used. If it's going to be a strict, healthcare application, for example, for a regulatory submittal, it's probably not something that is going to be very conducive to just a computational approach. But if it's something like screening multiple chemistries, looking at potential impurities, trying to derive a GHS classification on something that we believe to be relatively non-toxic, those are scenarios where the computational process alone may be more sufficient.
Yeah, okay. Right, I've got one more question here for Pilar, which is from somebody who's looking for some latest information regarding toxicity test substitution for the eye.
For
that's what it says, but this is actually something we've covered in a different webinar, I think, the eye irritation one. So, sorry, looking through here, I'm thinking that you need to look at the webinar series because we covered the eye irritation in a previous webinar. And there was another question, actually. I think we might be able to answer this outside the webinar, and it's somebody who's asking if the differences between QSARs, alerts, and read across. It says, what are the key differences between these approaches? I think that's probably, we can probably read a book on it. You can probably answer that succinctly to say something. I think we've actually, with our technical issue, confused you into not asking too many questions. So I'm just flicking out through the questions. I think we've pretty much. So unless anyone wants to take that last question, I think we'll jumping forwards to that. But I think we'll wrap up now. I want to obviously thank you for your patience with our technical problems. That made it through, and the recording, when it's done, will obviously have edited that bit out. So, thank you again. Obviously, I'd like to say a big thank you to our two speakers as well for being so patient with the technical problem. It's been about over 530 delegates that signed up to our event, so thanks very much for that. Just point actually, we do send a survey after the questionnaire, so please be gentle with us this time. But obviously your thoughts, constructive thoughts would be much welcomed. I think Amy already mentioned that the sixth webinar in this series is on the 14th of April, and it's going to tackle the zebrafish embryo acute toxicity test. So that's something to look out for. And that is all I have to say. So thanks very much again, and goodbye. Thank you. Bye-bye.
Thank you. Bye.